This video is a quick Key Stage 4 introduction to the idea of enzymes. So our title today is Enzymes. By the end of this, you should be able to state the purpose of enzymes, describe how enzymes work, name the enzymes associated with the digestion of carbohydrates, proteins, and lipids. So we should write out our notes in this way with the title across the top and the learning objectives down the left-hand column. Then in your notes, you'll write the answers corresponding to those learning objectives. Finally, at the end of the lesson, you'll summarize what you've learned in your own words. So enzymes catalyze reactions without being used up in the process. Catalyze means to speed up. So in this case, we have a digestive enzyme. We have a molecule which has been broken down into smaller pieces. An enzyme will speed up the rate at which the molecule is able to break into smaller pieces. So the first thing we need to know is the parts of an enzyme and substrate. So the food is the substrate when we're talking about digestive enzymes. So whether that is starch, protein, or lipids. Next, we have the active site on the enzyme. That is where the breaking down takes place. That is where the substrate locks in with the active site in order to be broken down. And finally, we have the enzyme. The enzyme is a very large protein molecule designed to do a specific job, such as break down carbohydrates or break down proteins. We'll start with amylase. Starch, in this case, is the substrate. Amylase is a type of carbohydrate. That is an enzyme that breaks down carbohydrates. So you'll notice that enzymes which break things down have ASE at the end of their name. So, when starch is in the presence of amylase, the starch locks in as a substrate with the enzyme at the active site. The enzyme breaks the starch down into two smaller pieces. Next we have protease, again ending in ASE. In this case, protease is the enzyme and protein is the substrate. So when proteins are in the presence of protease, the protease will lock onto the protein and break it into smaller pieces. Finally, we have lipase, which breaks down lipids. In this case, lipids are the substrate and lipase is the enzyme. When they lock together, it breaks off the fatty acids from the glycerol, leaving behind the large glycerol at the top left and the three fatty acid chains.